Good morning, good afternoon, good evening, everyone, wherever you are. Welcome to this new edition of the Worldwide Neuro-Developmental Forum. Our guest today is HongKui Zeng. Hi, HongKui. She's another one of these guests that had to wake up quite early today, 7:30 local time, West Coast, Seattle. Probably raining. I didn't even look, I didn't ask you about that, but no, okay.
Yeah, it's a nice day today.
Okay, it's a nice day. So, okay, I'll take that away. Sorry, guys. So a few words before leaving the full screen to HongKui and her science. So HongKui is at the Allen Institute for Brain Science. She joined that institution in 2006, became executive vice president, and very recently, last year, director of the Allen Institute for Brain Science. So you're all familiar with this institution, with this center. And HongKui really played an instrumental and central and driving force kind of role there in putting together all these beautiful single cell analysis and insights into brain connectivity and molecular identity that we've been seeing over the past decade or so. HongKui received her PhD in molecular and cell biology from Brandeis University, where she was there working in fruit flies, studying molecular mechanisms of circadian rhythm. Then as a postdoc fellow at the MIT, she studied the molecular and synaptic mechanisms underlying hippocampus-dependent plasticity and learning. And her current interests, as I just alluded to, is understanding neuronal diversity and connectivity, more specifically in the mouse visual cortex. There's also work going on relating to humans. I don't know if we'll be hearing about it today. And I think, one of the hallmarks of HongKui's work and of the Allen Brain Institute work in general is this combination, high throughput combination of molecular, anatomical, and physiological approaches that, again, not only provide fantastic resource for the community, it's very generously shared, but also direct insights into the biology of the brain, the cortex in mouse and humans. So HongKui, it's a pleasure to have you here. Thanks again for having woken up for us, and we're looking forward to your talk.
Thank you, Dennis. Thanks for that nice introduction. Let me share screen now.
Yep,
okay.
You're all set.
Good. Okay. Thank you. Thanks. Welcome everybody to this webinar. So the collective goal of neuroscience is to understand how brain works. This is a really daunting task, considering the extreme complexity of the brain with millions to billions of neurons and a similar number of non-neuronal cells in a mammalian brain, and the trillions of connections among those cells. To tackle this complexity, it's really essential to take a multi-level approach to try to understand the organizing principles of the brain at all these different levels. From basic units of the brain, the cell types, to the connectivity and wiring diagrams of the cells to form some functionally relevant circuit, to the behaviors and activities of individual neurons in an awake behaving animal, and finally to a computational understanding through modeling and theory to decode how the brain processes information. At the Allen Institute, we have also followed this kind of logic and set up a series of technology platforms to address questions about brain computation, and our primary focus at this stage is on the mouse visual cortex. Our largest effort, as you can understand, is focused on actually the first level to try to understand the diversity of the cell types and how they interact with each other, and use that as a foundation to tackle more complex problems. So why studying cell types? To understand any complex system, it is essential as a first step to create a parts list for that system, namely cell type composition for the brain in particular. Once we have a parts list, we can then, especially a molecularly defined parts list, then we can gain genetic access to those parts, the cell types, through marker genes, enhancer elements, and things like that. And those genetic access will allow reproducible interrogation of the different parts across different labs. This effort will also allow us to discover not only new cell types, but also organizing principles in terms of cell type diversity, and eventually helping us to understand other biological mechanisms, such as development, evolution, and the diseases, how brains are affected by a variety of different kinds of brain diseases. So given all this importance, how do we study cell types? We think that in order to gain a comprehensive understanding and creating a comprehensive list of cell types, it is essential that we take a systematic and unbiased approach and to investigate cell type diversity. That means that we should investigate making a lot of quantitative measurements of different phenotypic properties at different levels, such as molecular transcriptional level, physiological level, morphological, and connectional level, and then use a data-driven approach to classify the cells into different types. Importantly, we need to look for the congruency of the different properties in order to come up with more rigorous, robust, and unifying definition of cell types. So with this kind of understanding, at the Allen Institute, we developed a set of platforms to do single-cell characterization to define cell types. And then we also use cell type-based approach to investigate the connectivity and function in a particular circuit. So our front-end approach is to use the highly scalable single-cell transcriptomic method to profile hundreds of thousands to millions of individual cells and come up with a systematic transcriptomic taxonomy of the cell types in the brain regions that we are interested in. At the other end of the spectrum of our approach, we use a number of different approaches, anatomical and physiological, to investigate connectivities at both brain-wide scale and at local synaptic level, using both anatomical approaches at light microscopy and electron microscopy level, and using physiological approaches to look at the physiological and functional properties of the connections. We also, like many other labs, we also use in vivo imaging and electrophysiology to look at the activities of different cell types in awake behaving animals. How do we bridge this two very different types of investigation? We use two major approaches to do that. The first, of course, is to generate cell type targeting genetic tools, which include both transgenic driver lines using marker genes identified from the transcriptomic tree, taxonomy tree, as well as nowadays using enhancer elements mapped through the combination of single-cell transcriptomic data and epigenomic data to hopefully to be able to more efficiently target different cell types. The second approach is the multi-model single-cell investigation through patch-seq. In this method, we collect physiological, morphological, and single-cell RNA-seq information from the same cell. And then we use this triple modality information to do integrated classification of cell types. The cell type definition derived from this kind of study is really important in terms of serving as a Rosetta Stone in linking the very different types of data and information and investigation. For example, using the patch-seq approach, we are now able to correlate molecular signatures with the local morphologies of the individual neurons. And then the local morphologies of those cells can then be used to decode cell type identity in our electron microscopy connectomics data set. So then we can then assign molecular identities of individual neurons in the EM data and correlate them with their local connectivity patterns. Finally, the last approach we have been taking is to use spatial transcriptomics approach, such as the multiplex FISH. Using multiplex FISH, we are not only able to obtain the spatial distribution and proportion of the different transcriptomic types, but we can also use it as a post hoc method on the tissues that have gone through connectional or functional interrogation. Again, enable us to assign molecular identities to the cells for which we have obtained connectional and functional information. So today I will talk about 4 major new studies that we have completed recently in terms of using the set of technologies to understand the cell type diversity. But before that, I just wanted to say that this set of abilities, platforms have now been really integrated into a community-wide effort in Brain Initiative to characterize cell types in the entire mammalian brain. So we're now part of the NIH Brain Initiative Cell Census Network, in which we and many other investigators together throughout the world together used a similar set of approaches, as I just mentioned, to profile cell types in different parts of the brain across 3 major mammalian species: mouse, non-human primate, and human. And we collectively, we are able to generate molecular taxonomies across different species, spatially resolved transcriptomic taxonomies, integration of multimodal information together, and finally derive connectivity wiring diagrams of the different cell types. And a lot of work has already happened, and I think in the coming few years, we will learn a lot about the cell type diversity in the entire mouse brain, and I'm very, very excited about that prospect. But here, my talk will be mainly focusing on cell types found in the cortex. So here is just a brief introduction of the different major classes of neuron types in cortex. The first class is the glutamatergic excitatory neuron class, and it's divided into several major long-range projection types. The first one is the cortical projecting intratelencephalic neurons that are spread throughout all the different layers from layer 2, 3 to 4, 5, and 6. The second major type is the pyramidal tract or subcerebral or extratelencephalic projecting cells that are mainly located in the deep part of the layer 5, so layer 5 PT neurons. And the third major type of excitatory neurons is the cortico-thalamic projecting neurons that are mainly localized in layer 6. Through transcriptomic studies, we also find 2 other major types of excitatory neurons that are not shown in this review here, and that is the layer 5 near projecting or MP neuron type, and as well as the layer 6B subplate neuron type. So those comprise the major types of glutamatergic neurons in cortex. The other major class is the GABAergic inhibitory cells. They are mostly local projecting within the cortex. As you know, they have extremely diverse electrophysiological properties and morphological properties, especially in terms of their local axonal morphologies and projection patterns. They are also distinguishable by major neurochemical markers into a few major types that I'm sure that you are all very familiar with. So, as I said, our front-end approach is to use large-scale single-cell RNA-seq to provide an initial framework for cell type classification. We have 2 pipelines set up at the Institute. One is a SMART-seq based full-length deep sequencing approach. No multiplexing, each individual cell is sorted into a well or tube. And this kind of sequencing gives us extremely rich content of transcriptomics information for each cell. The second approach is the more high throughput 10x chromium-based droplet 3' end shallow sequencing approach. So the 10x approach, even though it gives us shallow sequencing, it is extremely multiplexable. So it can be easily scaled up to thousands to millions of cells. In our first set of published studies, we profiled cells using the SMART-seq approach in both mouse cortex and human cortex. In the mouse, we studied two different cortical regions together, and in human, one. We find that cell types, cortical cell types in both mouse and human are organized in a hierarchical manner. And at the higher branch level, at the major branch and major cell type level, the cell types are very well conserved across the two different species. The difference across species mainly comes from the leaf branch or leaf nodes level of the hierarchical tree, as well as the individual differential gene expression that are present in all different types. So even though there's conservation for all the major types, we can observe individual differential gene expression across all these different types as well, which may support species-specific function. Across different cortical regions, how do cell types vary? So from our two cortical study, from the primary visual in the mouse, from the primary visual cortex and anterolateral motor cortex, those two cortical areas at the extreme end of the cortical sheet, we found that GABAergic inhibitory neuron types are completely shared across those two different regions, whereas the excitatory neuron, glutamatergic excitatory neuron types are highly distinct between those two different regions. The distinctions not only reflected at the number of cells contributing to each transcriptomic cluster, but also the number of differentially expressed genes for each cluster, shared cluster, or pairs of highly homologous clusters between glutamatergic and GABAergic neurons. So there are a lot more differentially expressed genes in homologous clusters on the glutamatergic side. So this is the profiling of just two cortical regions. What happens if we have profiled cells across the entire cortical sheets? And what happens in the higher associational areas of the cortex? So recently, we completed another much larger scale transcriptomic study in which we profiled all the regions in the mouse isocortex or neocortex, as well as all the regions in the mouse hippocampal formation. So for isocortex, we profiled all the sensory motor cortical areas, as well as prefrontal, medial, and lateral associational cortical areas. For hippocampal formation, we profiled not only the hippocampus itself, but also the subbigular complex that's surrounding the hippocampus, as well as the lateral and medial entorhinal cortex. So this really comprehensive study allows us to assess cell type variation across the entire neocortical sheet, as well as to perform a systematic comparison of cell types between a neocortex structure, isocortex and neocortex, and a evolutionarily more ancient, considered a more ancient structure, archicortical structure, the hippocampal formation. We used both 10x and SMART-seq approach to profile all those regions, and we used a computational approach to integrate the cells from the two different methods together to generate this very large-scale consensus taxonomy tree. So as you can see here, the taxonomy tree dramatically expanded compared to our previous two cortical area study, and that expansion mostly occurred on the glutamatergic excitatory neuron class. So what about the principles we identified, the rules were identified in our previous study? Do they still apply here? We see that they do apply to some extent, but there is also extension. On the GABAergic side, we find that most of the cell types are still shared across all the different regions, as shown by this rainbow color indication of where cells come from, the regional source. But at the same time, as you can also see, obviously, that there are a bunch of cell types, the new types that we identified here now that are specific to certain regions. And those cell types are all colored in green, which are regions belonging to hippocampal formation. So we found that in addition to the shared GABAergic types across all different regions, there is also a unique set of GABAergic cell types that are unique to the hippocampus and its neighboring retrohippocampal area. On the glutamatergic side, we see that there's still a lot of regional specificity of each cluster or type. At the same time, we also now see that there are many cell types that are shared among different regions, and in particular shared among different neighboring regions, suggesting that the picture may be more complex than what we have seen before with two cortical areas. And we're gonna investigate this part a little further in the next couple of slides. First, we want to compare the glutamatergic cell type composition between hippocampal formation and isocortex. And here shows a UMAP of all the glutamatergic cells. This UMAP is colored by subclasses, higher branch level organization of the taxonomy tree, and this one is colored by regions. As you can see that there are some major continents here that are composed of cell types coming from all kinds of isocortical or neocortical regions. And there's an extensive overlap of the cells among those different cortical areas. Besides those major continents are really divided based on their major types of projection neurons. This big part is the IT cells that I talked about earlier, the intracortical projecting neurons from layer 2-3 to layer 6. And this part is the cortical subcortical projecting layer 5 PT. And this major branch here, it contains the layer 6 corticothalamic neurons, the layer 6B subplate neurons, and also the layer 5-6 MP neurons that I mentioned. So surrounding those major continents of isocortical neuron types, you see that there are many, many tiny islands of cell types that all come from hippocampal formation. So, so glutamatergic types from hippocampal formations are highly distinct from the isocortical ones. At the same time, they are also very related because of the location. All these different hippocampal neuron types are strategically located in different parts of the UMAP that have some kind of a relatedness with their nearby isocortical neurons. So then we did a more direct search or mapping of all the hippocampal formation clusters with mapping to the isocortical neuron types. And we aggregated those mapping into isocortical subclasses. And we find this kind of very, very nice, actually highly significant one-to-one correspondence between different hippocampal formation clusters and specific cortical types, suggesting to us that all the major types that we have found in cortex also exist in hippocampal formation. So the hippocampal formation has the same level or maybe even high degree of cell type complexity, a similar level of cell type complexity compared to the cortex. And we can find corresponding IT, intracortical projecting neurons in both superficial and deep layers of the different hippocampal formation regions, such as entorhinal cortex and post pre-para subiculum. We can also find deep layer corticothalamic projecting and the layer 6B subplate neurons in different parts of hippocampal formation as well, including entorhinal cortex and subiculum. And most significantly, we found that the subiculum and CA1 neurons in hippocampus actually have the highest homology with the layer 5 subcortical projecting PT neuron types. I'll come back to this homology later in my slides. And here I just wanted to show you another major finding from our study, which is really a large-scale gradient-like continuous transitioning of cell types in both isocortex and hippocampal formation or hippocampus itself. The first type of gradient transition is vertical. And this is looking at all the intercortical projecting IT neuron types from layer 2-3 to layer 4-5 to layer 5 to layer 6. And we found that all these different types or clusters are distributed along the cortical depth in an almost continuous manner without clear broader distinctions. So a large number of transcriptomic clusters distributed across the entire cortical depth in a gradually transitioning manner. This is a vertical gradient. At the same time, we also observed extensive horizontal gradients of glutamatergic types across the entire cortical sheet. So the entire isocortical sheet is shown here in a flat map with sensory motor areas arranged in this kind of anterior-posterior dimension, and then with higher order associational cortical areas mainly located in the prefrontal part, the medial part, and the lateral part. The gradients are shown here for each major subclass, as I mentioned earlier, from IT to PT to CT and MP and layer 6B. As you can see here, the cells from different regions, as color-coded here, are all gradually transitioning throughout the landscape. We find that the gradient in the anterior-posterior dimension is very continuous, whereas the gradient in the medial lateral dimension is more discrete. And as you can see here, actually a number of the isolated clusters, such as here the colors showing anterior cingulate and retrosplenial clusters, are clearly more distinct from the other parts of the cortex. So the medial lateral transition is much more discrete with the medial associational areas and lateral associational areas more distinct from other cortical areas, whereas the anterior posterior dimension and the transitioning of cell types is highly continuous across all the major subclasses. And there are some distinction here as well between the different subclasses. One particular thing I want to note is that the subcortical projecting layer 5 PT neurons are appear to be much more discrete and regional specific, as you can see here from the UMAP, compared to all the other major subclasses, suggesting that this group of neurons may have higher regional specificity, whereas many of the other cell types are distributed continuously across different regions. Similarly, we also find a multidimensional gradient distribution of cell types in hippocampus, in particular CA1 and CA3, as well as its neighboring subiculum and prosubiculum areas. And those different, we identify 4 major dimensions of gradient distribution. One is proximal distal, the other is dorsal ventral, and then the third one is superficial deep separation, especially in CA1 and CA3. And then the fourth is actually an activity-dependent gradient, whereas in which some clusters exhibit high activity level, meaning high immediate early gene expression level, whereas many other clusters exhibit low immediate early gene expression. So previously, a lot of studies have already shown that cell types in the hippocampus itself are not uniformly distributed, and they form various gradients. However, the exact nature of those gradients was not entirely clear, and was actually in much debate. Because of the studies are isolated, piecemeal kind of studies. So through this kind of a comprehensive study from our large-scale transcriptomics, we are able to reveal those 4 dimensions of gradients all at the same time, and with each carried out by a different set of genes that we are able to identify from our data set. So demonstrating really the power of using this kind of a comprehensive approach to resolve the complexity in the tissue. So in summary, for this part, for this study, we developed this diagram showing all the cell types, major types, we call it super types, identified across all different areas in isocortex and hippocampal formation. And there are this diagram really illustrates several major findings. The first is the parallel cell type organization between hippocampal formation and the cortex. We are able to find new cell types that were not known before in hippocampal formation, different parts of the hippocampal formation that share similar molecular signatures and layer specificities as validated by a large number of in situ hybridization data, which I'm not showing here at all. That we find, a homologous set of IT neurons in the superficial layers of entorhinal cortex and subiculum, as well as a set of deep layer cortical thalamic projecting and subplate and near projecting type of cells in those regions as well. And importantly, we identify that subiculum and CA1 act as the output cell types of this entire hippocampal formation circuit. Maybe they may play a role similar to the layer 5 subcortical projecting PT neurons in the isocortex that send information out of hippocampus to a wide range of other cortical and subcortical regions. So a parallel organization between hippocampus and cortex, and suggesting that there may be those 2 regions may be co-evolved during the evolution, and they may share some kind of a developmental programs between the 2, informing actually highly similar overall circuit structure. The second major finding is the large-scale gradient distribution of cell types across many different isocortical areas. At the same time, I didn't mention much at all. We also identified a number of specific region-specific cell types, and those cell types are mainly found in the medial cortical part, in the anterior cingulate cortex and retrosplenial cortex, as well as to some degree in the lateral cortical part. But we rarely find unique or region-specific cell types in other sensory and motor and even prefrontal cortical areas. The existence of large-scale gradient distribution with some clusters preferentially present in some areas versus the other. Together, this landscape really paint a highly complex landscape of cell types in the cortex that are neither region-specific nor completely uniform. So we think that those cell types collectively together define a cortical modularity, different modules in the cortex, and they also help to refine regional specificity together with connectivity patterns of those neurons, which I will talk about later. Okay, so now I will transition into the second type of study that I want to talk to you about. And this is really a multi-model characterization of cell types by patch-seq. In another recently completed study, which was just published, we profiled more than 4,000 individual GABAergic interneurons in the mouse visual cortex. So this is just coming from one cortical area. And we are able to map the transcriptomes of all those individual cells into our transcriptomic taxonomy tree. And we find that, interestingly, those GABAergic neurons are also organized in some kind of a layer selective manner. Many of them are organized in a layer selective manner, and also many of them exhibit some kind of a continuous transition across different layers. And this is particularly obvious in the somatostatin and parvalbumin subclasses of GABAergic neurons. We are able to use also the electrophysiological properties to group cells into different major classes, although the resolution of the physiological properties is not as high as the transcriptomic, as the resolution transcriptomic data can give us. We also have local morphologies from 500 out of those 4,000 neurons that we have obtained EFS and transcriptomic data for. And using this data, we are then able to perform clustering or classification analysis between two major modalities. One modality is combine morphological and electrical information together to generate what we call ME types. And those ME types are completely independent from the T types that we obtain by mapping the transcriptomic profiles into our pre-existing transcriptomic tree. So those two independently derived classification correlate with each other extremely well, as shown in this diagram. And we were very happy to see that all the clusters are aligned on this diagonal line. At the same time, as you can also notice that this correspondence is not perfect. It's not one-on-one correspondence. There's a substantial degree of local fuzziness between the neighboring similar types, either transcriptomic types or morphoelectrical types, suggesting a differential variation at a more refined level of the hierarchical organization of the cell types. And then using this kind of information, using the mapping statistics, we tried to derive what we call integrated cell type definition, which is the MET types. And we find that the MET types show much greater robustness and consistent profile properties, higher degree of correspondence between transcriptomic properties and morphoelectrical properties. So here shows the 28 different MET types that we defined through this study for all the GABAergic interneurons in the mouse visual cortex that covers all the major subclasses. The importance of defining the MET types is that the degree of congruency across different modalities is substantially improved, and the mutual predictability from molecular signatures and morphoelectrical properties is also now much higher. And this mutual predictability is important in bridging different types of data together, as I mentioned earlier, for example, in trying to assign molecular identities in, for example, electron microscopy connectomics data. So we think that the MET types would be a way to provide a Rosetta Stone for linking different kinds of structural, molecular, and functional studies of cell types. So this is GABAergic interneurons. What about long-range projecting cortical excitatory neurons? So we also have a separate project, it's a single-cell projectome project, in which we obtain full neuronal morphology reconstructions in a brain-wide manner. through a pipeline that we co-developed with a number of collaborators, especially several different groups of collaborators in China. We developed a genetic sparse labeling approach to label single neurons in a sparse and strong manner. And then we collaborated with the group in China to use the FMOST imaging method to do brain-wide, high throughput, high resolution fluorescent imaging of the sparsely labeled brains. And then we developed a computational platform to reconstruct morphologies of a single neurons from the FMOST data set. And we also register the FMOST data into our Allen mouse brain common coordinate framework, a 3D reference atlas, to allow anatomical annotation of the morphologies. And then combining the single cell morphologies with the registration together, then we're able to obtain quantitative information about projection patterns of individual neurons and use that information to perform classification based on the brain-wide morphologies of the neurons. And here I'm just going to show you some examples of the reconstruction and classification that we have done on several major cortical glutamatergic neuron types. The first is the cortical layer 2, 3, 4, and including 4 IT neuron types. And the second is the layer 5 PT neuron types. In both cases, we observed extensive morphological diversity or projectional diversity at the individual neuron level. So this shows an example of 10 individual layer 2, 3 IT neurons, all coming from primary motor cortex. And so you can see that each cell projects to a different set of targets, as shown in this summary plot, which is the projection map of the individual cells. Each row, each blue row indicate an individual neuron, and the orange rows here indicate the population level projection mapping that we have done before. So each layer 2, 3 IT neuron selects a subset of targets, and such selection appears to be random at the current stage. They do not correlate with the depth of the neurons of the individual cells. And as I showed before, the transcriptomic types are organized according to the cortical depth. So here, we don't see a correlation, strong correlation of the projection patterns with the cortical depth, suggesting that the projection pattern may not be strongly related to their transcriptomic profiles. Similarly, for the layer 5 PT neurons, we observed a wide range of diversity. Here is, again, showing just a few example neurons from the motor cortex, both primary and secondary motor cortex. From the morphological classification study, we can see that there's a major division of the layer 5 PT neurons. One is medulla projecting, the other is non-medulla projecting, or mainly thalamus projecting. And those two major divisions are very consistent with what we knew before, what we published before in an earlier study, showing that medulla projecting and thalamus projecting layer 5 PT neurons correspond to two major transcriptomic types that we identified. So that part is highly consistent. But then beyond that, we also observe additional morphological diversity that are not correlated with transcriptomic types yet. So, so this is a second example of extensive morphological diversity. The third example is the diversity in yet another cortical transcriptomic type, and this is what we call the CAR3. This is a very special intracortical IT neuron type that contains layer 6 neurons from many different lateral cortical areas, as well as major excitatory neurons from the claustrum. And this is a transcriptomically very homogeneous cluster. From this cluster, we observed that the individual cells, in both claustrum and cortex, exhibit extremely extensive projectional or morphological diversity. And this diversity can be grouped based on their topological, in a topological way, based on the soma locations of the individual cells from the anterior to posterior part of the cortical sheet. So the projection patterns here appear to be following a regional specific and topological rule rather than distinguishable by transcriptomic profiles, at least at the adult stage of the transcriptomic profiles. So somewhat contrary to what we have found for the GABAergic interneurons, we see that in the glutamate excitatory projection neurons, a much greater degree of morphological diversity than what can be explained at the adult stage transcriptomics profiles at this time. Okay, so finally, I wanted to use 2 slides to show you the last type of study that we have done in collaboration with our colleagues, a number of colleagues within the Brain Initiative Cell Census Network. Here is a combined study of single-cell transcriptomics and single-cell epigenomics. Again, we use the motor cortex, primary motor cortex as an example, for which we collected 8 or 9 different individual single-cell transcriptomic or single-cell epigenomic data. And we use the computational approach to be able to combine all the 8 different data sets using a variety of different sequencing methods together into an integrated taxonomy. And as you can see here, that the cells coming from each of those 8 different data sets are mapped into this integrated taxonomy very well. The importance of this study is that then now we can combine the epigenomic landscape together with transcriptomic gene expression profiles, combine them together in a cell type specific manner. So all the data are now integrated according to the cell types, integrated cell types we identify here. And we are able to correlate chromatin accessibility peaks with the neighboring or far away transcript expression in a cell type specific manner. And that allows us to identify specific enhancers or putative enhancers that we can use to target cell types using AAV viral vectors. So here I just show you some examples of the enhancer-based AAVs that we have generated, targeting major excitatory neuron types as well as major GABAergic inhibitory neuron types. We think that this approach is really tremendously promising right now because there are so many individual chromatin accessibility peaks or putative enhancer elements that can be easily identified for the different cell types in the data that we can now test and further develop. So in conclusion, I have shown you that large-scale single-cell transcriptomics provides a framework for cell type classification. We find that the different cortical regions have shared interneuron types. However, hippocampal formation also has its own unique set of interneuron types. We find many highly distinct excitatory projection neuron types in both cortex and hippocampus. Some of them are highly region-specific, especially in medial and lateral associational cortical areas, whereas many other types are distributed across multiple regions in a gradient manner. We also observe gradual but discrete transitions of cell types between neocortex and retrohippocampal regions. Multimodal classification refines the cortical interneurons into MET types, the unified MET types that have higher degree of congruency across the different modalities and a higher degree of mutual predictability. On the other hand, comprehensive brain-wide single neuron morphology reconstruction reveals extensive morphological and projectional diversity, even in relatively homogeneous transcriptomic types. And what I didn't mention you here is that our morphology reconstruction also includes neurons coming from different cortical regions and that belong to the same transcriptomic types. And obviously, those neurons from different cortical regions have region-specific long-range exon projection patterns, even though transcriptomically they belong to cell types that are shared across those different regions. So there's another degree of diversity that cannot be explained by transcriptomics yet. Finally, I showed that combination of transcriptomic and epigenomic profiling enables enhancer discovery and the generation of novel cell type targeting tools. And overall, there's a lot. I just give you some highlights of what we have found in our large-scale data generation across the entire BICCN consortium. And there's really a lot more that we are learning from our data. And we found that this kind of comprehensive multimodal characterization of cell types, even from just one cortical region, will allow us to integrate transcriptomic, epigenomic, spatial, physiological, morphological, and connectional properties all together, enabling bridging analysis in different modalities and as well as across different species. So with that, I want to thank a large team at the Allen Institute, including both senior and junior investigators who are leading the various projects that I told you about. It's really this kind of a collective team effort making this a tremendous scope of work to happen. I also wanted to thank our founder, Paul G. Allen, for his incredibly generous support and his vision and encouragement, and also a number of grants that we have obtained from NIH, especially through the BRAIN Initiative. With that, I want to thank your attention. I didn't talk about development at all. This is all about adult stage cell type diversity. But I think the roots of a lot of those diversities probably come from development, and I'd be happy to take questions from you guys. So I will stop sharing now.
Thank you very much for this beautiful presentation, HongKui. Those people were awfully close to one another on that picture. Our standards have changed. Yeah, it's like you said, it's an impressive teamwork, team effort, and yeah, thank you for providing us an overview of that. A few questions. I'm going to read them out loud for you and leave you the time to think about them and answer them. First one is from Seth Blackshaw. How many very rare but distinct cell types are present in the cortex, and what would it take to identify them all? How many more cells do you need to profile? So I guess a power analysis kind of question.
Yeah, I think we have pretty exhaustively identified cell types in the cortex. I'm not sure. I mean, we can talk about abundancy because we have profiled over 1.3 million cells with cortex and hippocampus combined together. And we feel like we are able to recover cells that are present at maybe 0.01 abundancy. We should be able to recover them. And so, and I'm not sure how much we would care about the cells types that are even below that abundancy. But from that, we do identify some cell types that are highly specific to certain regions. There are also several GABAergic interneuron types that are rare but very interesting.
So what's your view on how the complexity of different nervous systems and cross-species comparisons reflects the existence of additional cell types? I mean, there's some data out there comparing species. You've alluded to it too. But globally speaking, what's the importance of cell types in determining the complexity of a given nervous system?
Right. So I think we've done comparison between human and mouse, and a surprise finding here is that we don't see cell types in the human cortex are more complex. Cell type composition in the human cortex is more complex in the mouse cortex, right? So that's a big surprise to us. So where is the complexity or sophistication of the human brain coming from? I think it, different people have different, it could be still, the issue could still be technical, meaning that maybe in human, we have not done as good a job as what we have already done in the mouse. But it's also possible that that is just the reality. If you look at a single cortical region, the cell types are not that different across species. However, what it matters is the size of the human brain and the fact that there are a lot more different cortical regions than in the human than in the mouse. The number is comparing between 30 and 180, something like that. And
as defined how?
As defined how? I think as defined by function, right? Maybe connectivity and function. In human, cortical regions are defined by functional imaging, fMRI and other kind of functional imaging, which is supposed to represent networks. And I actually believe that it's also defined by cytoarchitecture, which is maybe the relative, maybe not in terms of the cell types themselves, but it's just the relative proportion of the different cell types. And I think the connections, inter-area connections, are maybe more important in terms of defining cross-species difference. Mm-hmm. And as I said in my talk, inter-area connections are not fully reflected in transcriptomics yet.
Yeah, we talked about that. Yeah, I do think there's some data out there also showing that unbiased analysis of human cortical histology highlights different areas histologically, like you were saying. It's not only not only in our mind's eye. Okay. Yeah. Another question is there's related work of Seth Grant, which he dubs synaptome, mapping synapse proteins across the cortex, but also across an animal's life. In your data sets, is there a distinction of synapse proteins and specification of age? So I guess, temporal aspects, which you might not have yet, but spatial aspects?
Yeah. So that's a very interesting question that we should look into. Cell types, there are definitely, that question here. We can
just erased it, sorry.
You just erased it. Yeah, we can definitely identify many different synaptic proteins, and we can derive cell type specific synaptic protein profiles. I'm not sure if they will correlate with the study that Seth Grant has done, because in his study, he is not able to distinguish, he's not doing single cell, and he's not able to distinguish between different types. It's really, all the synaptic proteins combined together. So I don't know how well that study correlates with our cell type based analysis yet, but from our study, we can definitely derive cell type specific gene networks for both synaptic proteins or ion channels or transcription factors, all those different gene networks. We just, there's a lot to learn from our data. We just haven't had time to do that yet. And of course, it's extremely important to look at across ages. This is something that we are just beginning to do, but we also hope that the field, like Dennis here, could do a lot more in that area as well.
So HongKui, what's your gut feeling, your intuition about the importance of extrasomatic transcription and of the proteome? How much, how many additional dimensions of information are lying there, and to which extent could that account for differences that we're not detecting? What's your feeling about that?
Yeah, so I'm not sure about extrasomatic transcriptomes. right? The transcripts that are, I don't know if that's what you meant, the transcripts that are distributed maybe in the
Yeah, that's what I meant.
dendrites
I mean,
and axons. Yeah, we there, there's no way we can recover them now using the existing transcriptomics method, right? What we are profiling is really just somatic transcriptome, and we lose the information. So the diversity in the dendrites of transcripts in the dendrites and axons, unfortunately we have no idea about.
what do you think? Would you take a
What do I
guess?
think?
Yeah, do you think
What do
like
I
there are some key thing that we're not seeing, and that in 5 years or 10 years from now we'll say, well, no wonder we couldn't figure it out because the key is there.
I'm not sure. I'm not sure. I mean, they
It's
are now
recorded,
very
right? You'll be on
advanced.
YouTube in 10 years, so
Okay,
me
you
too,
check.
by the
Yeah,
way. No,
yeah. Check my video. Check my answer here in 10 years. See, I'm right or wrong.
okay, might even be in 2 years. Who knows?
Yeah,
Yeah,
maybe even
and proteome, the importance of
proteome. I
translation.
think proteome definitely is something important, very important. It's again, unfortunately, we don't have a good way to profile proteomes at a single cell level. That's a major technology barrier to us. But I strongly believe there will be protein level of diversity that's not reflected in the transcriptomics yet. Absolutely, yeah, and that may be a bridge, between transcriptomics, gene expression, and the physiological properties, as well as maybe connectivity, right? The discrepancy that we have observed could be partially mediated by proteins.
Yeah. Okay, Shingo Nakazawa, thank you for the beautiful talk. How do you think the difference of morphology among the same cell types emerge?
Yeah, that's a great question. I think we've talked about it a lot. I think there are three possibilities, all maybe related to development. So everybody was saying that, okay, you don't see that correlation in the adult state. You might see that in so there could be three different developmental mechanisms for generating morphological diversity that we see in adults. One is that there is a genetic program in adults. The molecular, maybe Dennis, if you could mute yourself, that would be
Sorry.
helpful because I see echo of myself,
Sorry
and
about
that's
that.
a little distracting. Yeah, no problem. One is that there could be more extensive molecular diversity during development that correlate with the morphological diversity, right? So there is a genetic program existing in development, but kind of disappeared in adult. That's one possibility. The second is that it could be really activity-dependent mechanisms that shape the morphological diversity, and that activity-dependent mechanism may not rely on an intrinsic genetic program. The third is it could just be a stochastic. It could be that a set of neurons, layer 2-3 IT neurons, they are transcriptomically homogeneous. However, just for stochastic under a stochastic mechanism, each of them chooses a random subset of projection targets, and it's the population coming together reflecting that type level identity. So
Yeah, food for
very
thought.
different possibilities.
It's a provocative thought. I mean,
Yeah.
it's hard to disprove a stochastic event, right? But sure. Yeah, I have nothing to say against that. Yes, I agree. We talked about it offline. So, yeah. A question from Yorick Guiton. Your paper, Brain-Wide, indicates use of both male and females. Do your data further display relevant differences across sex?
Yeah, so our data include a male female balance. So in almost every cluster that I showed, it includes both male and female cells. Maybe just a couple that has only cells from one sex, but we're not sure about the validity of that finding. So from what we have seen now for the cortex and hippocampus, we don't find sex-specific clusters yet. However, that doesn't mean that there is no gene expression difference within the same cluster between male and female. we have not investigated that yet. And also, that doesn't mean that there's no sex-specific clusters in other parts of the brain. So it's very likely. And I think we've already now had preliminary data from other parts of the brain that has sex-specific clusters.
Yeah, I think something recently came out. Maybe somebody in the audience remembers. I saw something on Twitter recently on, I think, some hypothalamic region, or maybe somebody knows. If you remember,
hypothalamic region, for sure.
Last question, perhaps. Is it possible to use specific enhancers identified for lineage tracing of neurons during development and in neurological disorders?
Yeah, so that's another great example. So we show that even using adult stage transcriptomics and ataxic epigenomics data, we are able to identify specific enhancers. And it will be very interesting to extend that kind of work into development, right? If we can co-profile RNA-seq, generate data for RNA-seq and ataxic together, we will, identify a lot more elements as well, and it will be very interesting to see how those elements, enhancers, change with time, in terms of their coverage and specificity. And sure that those identified at specific developmental time points will be useful for lineage tracing, for development, and for treating developmental disorders.
So I have a bit of a provocative question with regard to the enhancers. What do you think the enhancers tell us that we don't know from transcriptomics alone? I mean, in many cases, when they're done side by side or sometimes simultaneously, it's something that you would predict, right? The enhancer sites are accessible for the transcripts that you detect. But what's the added value there?
Yeah, that's a great question. So far, for us, it seems to be that the enhancers are still mainly confirmatory. And the data that are being generated now is not as rich in content as transcriptomics, though they're not giving us additional information. The ataxic is very shallow. The number of reads per cell is very shallow. It doesn't give us that much information yet, but I was thinking that maybe once we are able to generate a lot more data from a lot more cells, when the data become richer, maybe additional patterns will emerge. One very interesting question I would like to know is that whether chromatin accessibility peaks or regions precede the emergence of those regions, precede the emergence of transcription or transcripts, right? So does it provide any predictive values about what may be happening in the future? That's a developmental, timing kind of question. We also know that so there is also more epigenetic marks, landmarks, kind of retained in the adult stage. So those landmarks may tell us more about the developmental origins of the cell than the transcriptomes, can tell. That's another possible additional information that we may get. I think this all depends on really advanced computational analysis of the data. I feel that now all this omics data are so rich in information that we really only have scratched the surface in terms of what we can extract out of that. I think, with more advanced analysis method, we will be able to actually extract more information and perhaps even insight from the data. For example, for epigenetics, I would really love to see, can we begin to identify regulatory patterns? Right, with all those peaks identified and the peaks corresponding to different cell types, and in the future corresponding to the timing during development, I really hope that actually more can be learned.
All right, that's a good conclusion sentence. Let's hope more can be learned, and I think you guys are in the pole position to help us understand that. So thanks again. HongKui, was a pleasure listening to you and discussing. Pleasure to have you here. Thanks to all of you that are on the other side of the screens for attending. Have a good end of the week, and take care, and see you sometime soon.
Yeah, hope to see you guys in person soon. Thank you for the opportunity. It was fun.
Bye-bye.
Bye.